Hello and welcome to our group's presentation for the Micro 405 group project. Our project aimed to design an extracellular bioenzymatic complex in microbial organisms capable of generating biofuels. Biofuels can be produced from plants as well as agricultural, commercial, domestic, and/or other industrial wastes, and are mainly used for transportation. They currently provide 3% of the world's transport and will become increasingly in demand as we transition from fossil fuels to renewable energy sources. Lignin valorization is one of the methods used for producing biofuels. Lignin is a highly branched heteropolymer composed of phenylpropanoid units, and it accounts for 18 to 40% of plant cell walls. It is one of the most abundant renewable resources and the only source of renewable aromatics on Earth. Unfortunately, it is severely underutilized, as effective lignin valorization would yield more than tenfold added value than just burning it for energy production. However, there has been a promising attempt in recent years to harness the power of microorganisms capable of processing these aromatic compounds to produce more useful chemical compounds. We focus our project on this paper by Wu et al., as Melina will describe in the next slide.
Our project originated from a paper from Wu et al. Published in 2017. Its aim was to genetically modify E. coli to express the enzymes necessary for transforming vanillin, a product of lignin depolymerization, into catechol, which is a versatile molecule used in the synthesis of a wide range of polymeric materials and high-value chemicals, including biofuels. They used a vanillin transporter for introducing vanillin inside the cell, as well as a vanillin-inducible promoter, ADH7, to turn on the expression of the enzymes. This vanillin-inducible promoter was selected to replace the commonly used PTRC promoter, which induces the expression in the presence of IPTG. However, use of the IPTG requires monitoring of culture growth and is a costly reagent, making it unfeasible for use at industrial scales. Unfortunately, when using the ADH7 promoter, production of catechol was greatly decreased, indicating that the expression of bioenzymatic proteins was weak, and there was a lack of strength of the ADH7 promoter. In addition, much of the catechol remained inside the bacteria, making it hard to retrieve the final product without destroying the cells. This also means that there was an accumulation of intracellular vanillic acid, resulting in high levels of toxicity. Therefore, our project seeks to optimize and increase the catechol yield for biofuel production without any accumulation of toxic byproducts. A paper published by Vorak et al. In October of 2020 used a genome-reduced strain of Pseudomonas putida to express an extracellular bioenzymatic scaffold capable of recruiting proteins into close spatial proximity. This strain, P. putida EM371, lacks all non-essential outer membrane structures, and hence is properties beneficial for extracellular recombinant protein production and facilitated downstream processing. P. putida is a popular choice for the biotechnological upcycling of polymeric waste due to its metabolic capability, safety status, rapid growth, resistance to inhibitory chemicals, and compliance to genetic manipulations.
The scaffold complex they expressed in P. putida, SCAF19L, consists of 3 cohesin domains attached to a peptide backbone capable of forming interactions with nearby Docker molecules, which can be added as tags to pre-existing proteins. These proteins assemble with the scaffold and get translocated to the plasma membrane via a monomeric type V secretion system known as an autotransporter, which is commonly used for decorating surfaces of gram-negative bacteria with recombinant proteins. Now to bring everything together. Our project outlined the design of a plasmid encoding for Dockerin-tagged enzymes responsible for converting vanillin to catechol, as well as the scaffold and autotransporter molecules. This plasmid will be transformed into the genome-reduced P. putida strain EM371, which will now express, assemble, and translocate the enzyme scaffold complex to its cell surface, allowing for catechol production to occur outside of the cell in a localized manner. The bacteria will be cultured in a bioreactor, where the vanillin will be added. We hypothesized that this system will both increase catechol yield and limit aromatic toxicity, optimizing the process for large-scale fermentations. Our workflow consisted of the following steps: a homolog search, ribosome binding site optimization of specific genes, PCR amplification of the promoter, scaffold, autotransporter, and bioenzymatic proteins, genomic assembly with the plasmid, transformation and expression into P. petita, and bioreactor design. The XYLS promoter and P7-238 expression vector were used by Dvorak et al. In constructing their extracellular scaffold in P. petita, so we decided they would be appropriate to use in our project as well.
First, a homolog search was conducted to identify enzymes potentially more efficient than the ones proposed for this experiment. The original organism, the new organism from which the homolog originates, and its GenBank code were summarized in the following table. The efficacy of these new enzymes would have to be tested in the lab before being selected to replace its respective homolog. The ribosome binding site optimization was performed using the OIT-Lab RBS calculator. The mRNA sequence of each enzyme was run through the predict translation rates algorithm to determine each enzyme's translation initiation rate in Pseudomonas petita. We then ran each enzyme's mRNA sequence through the control expression levels algorithm to optimize the ribosome binding site of each enzyme. Due to the larger nature of the operon, ribosome binding site strength was only optimized in the elements furthest away from the promoter to encourage uniform expression of all genes. Each of the elements used in the constructs were PCR amplified before assembling into the final construct. Proper primer design was crucial for correct assembly, so special attention was devoted to this step. Primers were designed to add an overlap region to each element corresponding to the adjacent fragment, and fusion PCR was used to add a Dockerin tag to each of the bioenzymatic proteins to allow for attachment to the scaffold. The plasmid overlap sequences designed for the first and last fragments of the construct incorporated recognition sites for the restriction enzymes used to linearize the plasmid in the genomic assembly process. The kanamycin resistance gene used for selection was already present in the P-SIVA-238 vector and was not included in this step.
The XYLS promoter, scaffold, and autotransporter were amplified via canonical PCR. Primers were designed to integrate overlap sequences that match the adjacent fragments to allow for proper assembly of the DNA construct. Specifically, the promoter was designed to overlap with the plasmid and the scaffold. The scaffold was designed to overlap with the promoter and the autotransporter. The autotransporter was designed to overlap with the scaffold and the optimized RBS of Lignin. To do this, the forward primer was developed by using the last 10 nucleotides of the upstream sequence and the first 10 nucleotides of the gene. The reverse primer contained the reverse complement of the first 10 nucleotides of the gene and the last 10 nucleotides of the downstream sequence. As was mentioned earlier, the bioenzymatic proteins required attachment of Dockerin tags for proper assembly with the scaffold via the cohesin domains, as well as addition of overlapping sequences with adjacent fragments. This was performed by fusion PCR, which required design of special primers capable of amplifying the desired final construct. In this fusion PCR example, we have Lignin. The same process will be done for the rest of the enzymes. The first and second PCR are the attachment of the primers in preparation for overlap PCR. The reverse fusion primer was determined by acquiring the complement of Lignin sequence, attaching it to the start of the Dockerin sequence. This will then be reversed so that it is going from the 5' to 3' direction. That will give us the reverse fusion primer. The fusion primer of the Dockerin is a combination of the complement of Lignin and the start sequence of the Dockerin. After this, the two products will go through another PCR that will anneal them. Another PCR is done to attach the primers that will make it ready for Gibson assembly. The optimized RBS sequences are to be used in the primers. The forward primers for this PCR is a complement sequence of the Lignin RBS that will end up attaching to the Lignin RBS sequence that is on the AG43 autotransporter and the start sequence of Lignin. The reverse primer is acquired through the same method as the reverse primer for Lignin. In this case, the Lig-M RBS and the Dockerin sequences are used. The 4 primers presented here are not all the primers used in the experiment, but they are the ones used to connect the necessary sequences, attaching the Dockerin to the enzyme and attaching the optimized RBS for proper assembly.
In order to assemble our final construct, we first cut the P-SIVA-238 expression vector, as well as the XYLS promoter and final gene arrow Y with the SAC-I and KPNI restriction enzymes. This allows for the promoter and the final gene segment to ligate into the open plasmid, preventing plasmid recircularization. To assemble the remainder of our genes, we chose the Gibson method due to its simplicity and readily available kits. Our pre-designed overlapping sequences for each gene will be chewed back in the 5' to 3' direction by a temperature-sensitive T5 exonuclease. This will create overhangs that will then anneal to the complementary overlap sequence found in the adjacent fragment. After annealing, the gaps will be filled in by a polymerase and a ligase, resulting in a complete circularized plasmid containing all of the elements in the desired order. Because of the unique overlap sequences introduced during PCR amplification, the final plasmid will only assemble in the correct manner. The plasmid can then be transformed into competent P. putida using methods such as electroporation. Successful transformants would be selected by adding kanamycin to the growth medium, and surviving colonies would be picked and sequenced to confirm the presence of plasmid containing our construct. Our newly modified P. putida would then be transferred to a stirred tank bioreactor harboring conditions for enhanced strain growth. This bioreactor was selected as it is optimized for large-scale commercial processes. In addition, the system can funnel out the liquid and separate our product from the main fluid, with the main fluid then being recirculated back into the tank. Vanillin would be added to the media within our bioreactor and transformed into catechol via the biosynthetic enzymes now expressed on the surface of P. putida. This catechol would then be able to be extracted from the mixture without disrupting the integrity of our cells, allowing for continuation of the process without creating a new cell culture. This catechol can now be used for valuable downstream processes.
Because this is a hypothetical project, there are several experimental conditions to keep in mind. We believe the main concern is assuring uniform expression of all genes, which can be accomplished by introducing variations in the elements that modulate expression. For example, RBS optimization can be extended to other genes besides the bioenzymatic proteins. We only used one promoter, but different promoter sequences can be introduced in other locations to boost expression. All the genes were introduced into one plasmid, but they can be split between several plasmids with potentially higher copy numbers. Finally, we opted for a constitutive promoter used by previously mentioned researchers, but it is possible that an inducible system would be more appropriate if the inducer is feasible and cost-effective. Homolog efficiency and bioreactor design will also play a role in the ultimate goal of increasing catechol yield. However, all these considerations can only be evaluated in an experimental lab setting. Ultimately, we believe our design introduces several key aspects that will aid in increasing catechol production for biofuel generation. By bringing the enzymes into close spatial proximity and localizing the reaction outside of the cell, we overcome the main limitations posed by other groups. We are excited about the advances being made in the field of lignin valorization and other biotechnological approaches to biofuel production as we finally transition to cleaner forms of energy.